Today we are going to discuss very important topic, that is gene expression. As we know, every somatic cell of our body has the same DNA, yet different organs of our body function differently. Why? For example, stomach has a particular cells like parietal cells that synthesize HCl, but the eye cell does not synthesis HCl. Likewise, alcohol dehydrogenase enzyme present only liver to metabolize alcohol, but the same enzyme is absent in neurons. It means every cell has the same type of DNA, but the genes of different but the genes are not activate in every cell. Like some genes that are expressed in particular liver cell but that are not expressed in neurons. In same cell, different times also decide when that particular gene be activate and produce particular type of proteins in proper amounts. It means gene expression is turning on the DNA gene to produce messenger RNA and proteins. First, we are going to discuss gene expression in prokaryotic cell. There are some basic differences in prokaryotic and eukaryotic gene expression. Number one, prokaryotic gene expression and eukaryotic gene expression. In prokaryotic, the DNA floats freely in cytoplasm, but in eukaryotic, the DNA they reside in chromosome in nucleus. Second, DNA transcription and translation that occur simultaneously in prokaryotic cells, whether transcription that usually that occur in eukaryotic in the nucleus, but the translation process that occur in the cytoplasm. So transcription precedes the translation. In eukaryotic, it means the gene expression at the primary level of transcription, whereas there are many levels of gene regulation in eukaryotic, at least 5 types. Number one, epigenetic level. Second, transcription level. Third, post-transcriptional level. Fourth, translational level. Fifth, post-translational level. These are the important difference between prokaryote and eukaryotic gene regulation or gene expression. First, we are going to discuss gene expression in prokaryotic cell. In prokaryotic cell, all the important functions that are bring about by a particular set of proteins that encoded together, that is known as operon model. It means the particular set of genes that encoded a particular set of proteins, that specific function, that all are encoded together, that known as operon in prokaryote cell. It means the particular messenger RNA formed from the particular set of genes that is a polycistronic transcription. First time to import this lac operon concept that is proposed by two French scientists. One is Jacob and Monod. They proposed lac operon concept first time in bacteria E. coli. Now we will discuss of lac operon concept. The DNA or sequence of lac operon has a two different part. One is called as regulatory region, that has a one portion called promoter. Second important, that is called as operator. And the second part, that is known as structural gene. This is known as structural gene. Lac Z, lac Y and lac A. This portion is known as structural gene, and this region known as regulatory genes. That have important two part. One is the promoter region of the DNA, where the particular the enzyme RNA polymerase binds. Second, operator, where activator and repressor binds. The E. coli that use blood sugar as their primary food, but whenever the blood glucose as a primary food, but whenever the level of glucose is low or absent, then the E. coli bacteria that able to digest lactose. It means when there is a full abundant quantity of glucose present, the lac operon that is turned off, switch off in phase, and they are not able to produce the particular enzymes, and hence the lactose is not able to digest in the E. coli. But whenever there is present of lactose and low level of and absence of blood glucose, then the lactose present in the medium that bind to repressor, and this lactose bind to repressor make it incapable to binding, make it incapable to binding to operator region, and so the RNA polymerase, so the RNA polymerase easily attached to promoter region. Hence, the structural gene that started to transcribe and later on translated into three proteins. One is beta galactosidase, second permease, and third trans acetylase. But whenever the lactose is absent and blood sugar, blood glucose is present, then this repressor easily that will bind to operator region and make RNA polymerase unable to attach to promoter region. Hence, the structural genes are not able to transcribed into messenger RNA. Hence, the further mechanism of translation that is blocked, and the important enzymes will not form, so they are not, the bacteria is not able to digest the lactose in the medium. Okay, now the second important, that is tryptophan operon model. अभी अपने लैक ऑपरन मॉडल पढ़ा, वैसे ही ट्रिप्टोफैन ऑपरन मॉडल है. इसमें भी डीएनए के दो पार्ट, जो ट्रिप्टोफैन ऑपरन है उसके दो पार्ट होते हैं. एक होता है रेगुलेटरी पार्ट, दिस इज कॉल्ड एज रेगुलेटरी पार्ट. सेकंड इज स्ट्रक्चरल gene. Tryptophan operon model, there are 5 set of genes. Tryptophan A, B, C, D and E. These 5 set of genes encoded together. In regulatory region, there are two type important region. One is the promoter, second operator, and third that is the inducer. It means whenever the tryptophan is present in the medium, then E. coli no longer needed tryptophan synthesis process. So this tryptophan operon model, that in the switch off mode. But whenever there is a absence of tryptophan medium, then these E. coli bacteria, they require to synthesize tryptophan. Okay, then the tryptophan lac operon model is switch on mode, and they synthesize the 5 different proteins that are very much important in synthesis of tryptophan in bacteria E. coli. For example, it is inducer that formed a repressor. But whenever in medium the tryptophan present, that tryptophan this is tryptophan bind to this repressor and make is liable to attach to operator. It means initially the repressor form that is not able to bind to operator region, it is called aporepressor. But when the tryptophan in the medium that bind to aporepressor and make it activate repressor by changing its configuration and it will easily bind to opposite region, and once it upper pressure and TRP that bind to opposite region, then the RNA polymerase not able to bind to promoter region, and the 5 set of genes, the structural genes that are not able to transcribe into messenger RNA, hence the 5 different type important enzymes and proteins not able to formed. It means lack of TRP, the operon model that will remain in switch off mode. But when in the medium the tryptophan is absent, then this repressor not able to bind to opposite region, hence the promoter region is bind to easily with the RNA polymerase and these 5 set of genes that form a messenger RNA, and 5 different important proteins, enzymes that will able to form, and they will very much helpful in synthesis of tryptophan in E. coli. It means gene regulation process that is conserve energy and space in our cells.